Okay. Right, so we have spoken quite a bit about objectives, and now we're going to I've introduced already the refractive index, and now we're going to try to figure out why we are talking so much about this refractive index and what are the consequences if we do not match the refractive index. So let's see. Right on the other side of me, yeah. So we're going to have a very nice workshop also afterwards about this. So this is what happens when we have a very good PFS. This one is not a real PSF. It is it's a generated PFS, and it's not me. It was Kurt on his macroscopy blog, which is excellent. So this is, you can say, a good PSF. It's symmetric. A point-spread function is never that symmetric. It's usually harder to see the rings on one side compared to the other. But anyway, and you can see because it's elongated like this is actually the side of the point-spread function, right? So this was acquiring an image through like a Z-stack of a bead and then displaying the Z-stack from the side. So this is good. It's symmetric. We can see the rings. We have a very nice little main lobe in the center. And when we get aberrations of any sort, what happens is it all goes down to hell. So we get like a much more elongated center of the point-spread function. And then we have, instead of rings, we have all sorts of shadows around. So it can be very different from one problem to another. Sometimes this one is still straight, but very often they are completely distorted like a banana. So when we have optical aberrations, the point-spread function is distorted. The focus may be different from the center to the periphery of the image. And imagine that creates a lot of problems in image analysis. We may also, as we have seen before, have different colors that are not focused at the same Z level. So that also is an enormous problem. So all these are optical aberrations that we want to avoid. And what is the thing with the refractive index mismatch? Because one of the way we can tackle these optical aberrations is to match the refractive indices of the sample and of the objective. So let's see what happens when we do not match. So I remind you that the refractive index is abbreviated N, and that is small n, and this is the refractive index. So when you see this, you will understand that this is the refractive index of the medium. So it means the capacity of this medium to bend the light compared to just traveling in air. So air is the reference, that's why it's n equals 1. So here we have an inverted microscope, right? The objective is pointing upwards and the sample is above. Here we have the objective lens, the front lens of the objective. And this is an oil objective. So on this front lens, we should put oil. And this is what we have here. So this is oil, and the refractive index is 1.52. You don't need to remember all these numbers. They are in the important equation paper. But anyway, there are only a few of them. So I think at the end of the course, you will remember them. So oil is 1.52. It's not always 5.2, so it can be a little bit different, but that's why we're using in this course. Glass is also 1.52. So what happens is that all these pieces, the objective lens, the oil, and the glass of the cover slip have the same refractive index, and therefore there is no refraction. If they have the same refractive index, light just travels undisturbed, okay? But then when the light reaches the sample, so the excitation light reaches the sample, then we have a different refractive index. In this sample, we have 3 little beads, fluorescent beads. One of them is flush against the cover slip, and the others are further away. When these beads start fluorescing, they act like a light source, and the emission light will go everywhere, but some of it will go back into the objective, and that's what we're looking at. So if we take a point-spread function, so we take a Z-stack basically of this little bead, we acquire a Z-stack of this little bead that is here flush against the cover slip. what we see is that we have a really cool point spread function. So we say z equals 0 because we are at the cover slip level. We have a really nice point spread function. If we now look at a bead that is a bit further away, and it's only 25 microns away, so you can say 25 microns is somehow the size of a not very big cell, right? And so just one, so you can say if you have like a cell, a monolayer on the cover slip, we're talking now about the top of the cells, right? About that level, you can see how distorted the light is. And if we go even further inside the sample, maybe 2 cell diameter away from the cover slip, it's even more distorted. This is what happens when we do not match the refraction indices. Sometimes your scientific question does not require you to go inside the sample, then that's fine. Sometimes your scientific question does not care too much. You can still analyze the images if the point spread function is distorted, but sometimes it's a big problem. So you have to be aware of the consequences of not matching the refraction indices. So we're going to have this really nice workshop afterwards that will demonstrate all this. So what can we do with the point spread function that is distorted like this? The main issue is that the distortion is different at different depths. So it's not the case that you can just set the correction color on the objective to something else and then image and it's going to be perfect. It doesn't work like this because you have an exponential effect as you go further away from the cover slip. So you do not have one position of the correction color that works. So manual correction of the manually moving the correction color does not work to correct for aberrations due to refraction index mismatch. The distortions grow exponentially with the imaging depth. You can correct if you use an automated correction color that grabs the objective and moves as you go deeper. That is possible. So what are the consequences of having this distortion? If we can see clearly that the PSF is elongated, so clearly everything will look more and more elongated as you go further away from the cover slip, but there's another consequence. So if we have a look here at how things work, so now we have an objective, and here we have the fluorophore is going to fluoresce in here. It's a little bit away from the cover slip, and this is a cell. So it's cells grown on the cover slip, and therefore this is like 1.33. It's the same refractive index as water. And here we have the cover slip, which has a 1.52 refractive index. So here we have refraction because we have a different refractive index. So the light that comes in from this fluorophore is going to reach the cover slip, and here we have refraction. That's normal, and this is exactly this effect, okay? And there is a little law like this that says that the bigger difference between these two and the more the refraction. Basically, that's what the law says. So if we have, the law talks about the angle, and is the angle with the vertical, what is like perpendicular to the cover slip. And so here we come in with a certain angle, and then afterwards we have a different angle, which is smaller. And let's say that this objective is a water objective. So we have water in here as an immersion medium. So what happens is that we're going to go back again from 152 to 133. And you see we had 133 here as well. So the refraction is going to be identical to this, but just reversed. So we have refraction again, and here we're going to have the same angle, but just after B. So what happens here when we measure how far this little molecule is from the cover slip, this is what the real thickness is. And here we're going to measure this thickness. It's going to be the same thickness, okay? Even though we have this little way. And that is, so because we have the same refractive index here and here, so here we're matching the refractive indices, the one of the objective and the one of the sample, even though we have refraction at the cover slip, because the angles are the same, we get the correct measurements. Good, so we get correct data in here. But now let's say that we use an air objective instead. So now we have a mismatch between the sample and the air objective. the air is basically the immersion medium, if you want to say. So in that case, the two angles are different. And we can see that there's a lot more diffraction in here when we go to air. And the measured distance, that the distance that is here, that is measured, is smaller. So our object, our sample looks compressed. When we look at the image, when we do a Z-stack of the sample, it looks shrunk. Okay, it's not, but this is what it looks because we did it optically. So we look like a compressed sample. Now, if instead we use an oil objective with a higher refractive index than the sample itself, we get the opposite. So now what we have is that because here and here we have no refraction because they have the same refractive index, it just goes straight. So what we are getting here is that we are measuring a distance that is much longer. And the sample is going to look elongated. So everybody has experienced this. When you go to your microscope, you put your sample there, you're using an oil objective, and then you look at the bottom of the cell, looks quite good. You look at the top of the cell, everything is elongated. That's why. Refractive index mismatch. Clearly, all the point spread functions are elongated, but the fact that the phenomenon is getting worse as we get away from the cover slip is because of refractive index mismatch. Okay, so if close to, if your sample is much better close to the cover slip than just a bit away, that's now you know. So how to know if it's going to be elongated or shrunk? It's quite easy. You follow the emitted light path. And if the refractive index increases, then the distances will also increase. So there is an elongation. So in here we have 133, and here we have 152. So the refractive index increases along the emitted light, and therefore we have an elongation. If the refractive index decreases, then we shrink. Okay? That's the way to remember, but it doesn't really matter. If you are at your microscope and you use an oil objective, remember that as you go away from the cover slip, if you see that things get worse and worse very quickly, it's because of that. Okay, and again, it's exponential. So what happens is not just that. So not only we get all this elongation that is happening, but this is another way of showing it. So here we have a water sample. So maybe like some cells in water, and here we're upright, right? Here we have an oil objective, and here we have water. So here we're matching the refractive indices, and here we are not matching. We're using an immersion medium that has a higher refractive index. And when we are at the cover slip, there is no distortion, okay? Because the light does not travel in the medium. It goes straight into the glass. So it's going to go straight into the glass, no refraction here, and no refraction here because it's oil and glass. It's going to go straight. So what happens at the cover slip is that we have no distortion. And as we go away from the cover slip, we get more and more distortion. So if you say this is like, this is what the sample is, what will happen is that the image of the sample is going to look like this. And if we do the same thing matching with using a water immersion objective, as we should, this is the sphere that we're actually imaging. What happens is that the image of the sphere is displaced, but it's correct because we have the same diffraction at the top and at the bottom. So where we image the sphere close to the cover slip or when we image it at the top, we get the same distortion, and therefore all the measurements are correct. So there's no distortion at the cover slip, but there's a lot of distortion away from the cover slip. And when we match the refraction indices, there is distortion at the cover slip and the same distortion away from the cover slip. So we have, and yes, I want to mention as well that this is the correct cover slip. If you do not have a cover slip number 1.5, which is what the objective manufacturers expect you to do, then you will introduce other aberrations that will pile up. So the cover slip is part of the design of the objective and the sample as well. So always choose number 1.5 cover slips. And also one of the issues that we have in here when we do that is that, as you can see from the top, when we image the top here, what happens is that we use only part of the objective. So we use only part of the numerical aperture of the objective. Remember, the numerical aperture is related to this angle, the cone of acceptance. But because of these optical aberrations, we only use a part of it. So we are, in practice, we're actually using a lower numerical aperture objective than we think. So maybe we think, oh, this experiment is very demanding. I'm going to use an oil objective. And what we're doing, because it's the highest resolution, because it has the highest numerical aperture, and what we do is that we actually decrease effectively. We use a decreased numerical aperture. That's not very good. Whereas in here, we use the full numerical aperture of the objective. Okay? So when we do not match, we use a lower effective objective. And that is because it is related to this angle. So when we use a smaller angle here, we do not use the full numerical aperture. So we have a smaller angle, lower numerical aperture, we have aberrations and an elongated volume. And in here, when we match, we have no aberrations, a correct volume, and a full numerical aperture. Okay? So please, if you have a water sample, if you have any cells that are in medium, if you have any cells in a gel, anything that is in water, or with the same refractive index as water, use a water objective, not an oil objective. Only use oil objective if your sample is in a mounting medium that has a high refractive index. Okay? So here is again, you will see it only when you look side by side. So here I have a sample on the slide, and then afterwards we put an aqueous medium, and then we cover with a cover slip number 1.5, and we image through the cover slip. So we have the mounting medium is between the cover slip and the sample, and we are imaging an aqueous sampling. We will see this next week. So we did our best, let's say, but the sample is not prepared very well. Because it's not on the cover slip. And here we use an excellent dipping lens, the 20x NA 1.0. It's a water dipping lens from Zeiss. This lens, is one of the white ones that I've shown you. It is designed to dip in the medium directly. There should not be any cover slip. These are the images that we're getting. They're quite nice, right? But if you compare now with another objective that is designed, it has almost the same numerical aperture. If this one is designed to be used with a cover slip, you can see how much sharper these images are compared to this, right? And this is because of the aberrations that are introduced by the cover slip. If you use a dipping lens, no cover slip. And you only see the difference if you look side by side, clearly. So the consequences of not matching the objective and sample refraction indices or using the wrong cover slip thickness are increased optical aberrations, reduced brightness, reduced resolution, and a loss of contrast, incorrect Z measurement. So I stress again, if you measure distances in Z, you absolutely must match the refractive indices, especially if you use a high numerical aperture objective. And so for a high numerical aperture objective, you actually decrease the numerical aperture objective when you do not match. This is one of the major causes of signal loss when we image thick samples, and both the excitation light and the emitted light are affected by these aberrations. And if you have a super thin sample and it's stuck against the cover slip, you have almost zero problem, because we have shown that the problem occurs as soon as you get away from the cover slip. But at the cover slip itself, we have no refraction. The aberrations they start appearing very quickly. Only a few microns away from the cover slip, you will already start seeing them. And the problem increases proportionally with steps. So if you have a tissue, the refractive index of tissues are usually around 1.4, a little bit higher, a little bit lower, depending on the density, depending on how many proteins there are in the tissues, how many lipids and stuff like this, how much lipid there is. So around there. So the ideal objectives are glycerol or silicone objectives with tissues. So how can we prevent optical aberrations and refractive index mismatch? I will say it again and again, please use the correct cover slips. And do not use cover slips with dipping lenses, and put your sample on the cover slip, not on the slide. So we'll talk a bit again about this in the sample preparation workshop. Adjust the correction color if your objective has one, and match the sample. And if you come to the workshop, if the objective that you normally use has a correction color, let me know and I can show you how to adjust it. And next year I will come around to making a video about it. And so match the sample and the objective refraction indices as much as possible. If it is not possible, use an objective that has a lower refraction index than the sample to use its full numerical aperture. So it is better to use an air objective with a water sample than to use an oil objective with a water sample. You want the refractive index of the objective to be lower or equal to your sample. And if the sample is touching the cover slip and is very thin, then any objective can be used because then we have no refraction. And tissues have about a refractive index of 1.4, so silicone objectives are good. Otherwise, you can clear them and match the refractive index. Live cells in medium are definitely best imaged with a water objective. So now I have a little example of things. Here, what do we have? We have a section of a tissue. It's labeled with 3 fluorophores and it's mounted in prolonged diamond. The sample is on the slide. And then with the mounting medium, the cover slip, we're imaging through the mounting medium with a 10x objective NA 0.45. And we're focusing on the red. Can anyone tell me if anything is wrong in this image? Yes, Maria?
It doesn't seem to be focused.
What is not focused?
Like different parts, the image is not in the same plane.
So that was a very good spot. Nobody ever finds it, so I'm very impressed. But look at this one. If now I display the image as we should, separated with the overlay and then each channel separated and in black and white, you can see very clearly that the red on which we focus is actually very well focused everywhere, but it's more diffused. Look at the blue. Yes, and this is, and if you focus on the blue, then the red is out of focus. So, and then it goes like rainbow one. If we would have like a far red, it would be even worse, right? The distance. So this is typical of aberrations. And this is chromatic aberrations. The blue channel is out of focus. And in this sample, the mounting medium was also autofluorescence. You cannot see it here. But yes, so please put your sample on the cover slip, not on the slide, and show each channel separately in black and white. This is the best way to display images. Okay, good. So do we have something else in the chat? Let me see. On the side, more spread the opposite side. Is the side